this session of our lecture series. I'm very pleased that we have Gustav Nilsonne with us. This lecture has been planned for a long time, and we thought when planning this lecture series that there must be one session at least about research data. And now in our lecture series, we have today's session about the assessment and the quality assurance of research data. And I will introduce our speaker and our discussant in a moment. But we will also have a lecture in January on data feminism, which certainly also touches on the topic of research data. And I think that we will also talk about research data in our discussion in February on the topic of open innovation. So today we have the pleasure to welcome Gustav Nilsonne, who was recommended to us very highly. And I will introduce him and our discussant or co-discussant, as we have fluent roles today, Sibylle Söring. Gustav Nilsonne is a trained medical doctor, but is a researcher foremost, with focus on neuroscience and translational research. His interest, and that is the relevance for today's lectures, include meta-science and investigating transparency and reproducibility and openness of medical research. He is a national Swedish coordinator within the consortium of the Swedish National Data Service, particularly for medical research. But moreover, he's also in the international networks of the Research Data Alliance, and he's also a task force leader in the European Open Source Cloud. And in 2017, he was appointed as the Open Data Champion by the Dutch Foundation Spark Europe. I'm very much looking forward to your lecture. Before that, I would like to introduce Sibylle Söring. We're very happy to have her among us as an expert. The expert on research data, as I would say, in Berlin, because she's the leader of the research data management team at the Freie Universität's University Library. During the last years, she has been involved in several research data projects and has led the development to the new research data policy of Freie Universität. Sibylle Söring is also a co-PI, so project leader, in a project funded by the Berlin University Alliance that aims at surveying the needs of researchers and formulating specific actions to foster the quality and openness of research data at the Alliance partners. So I think that we will have also some question to that. To link the discussion or the presentation of Gustav to the local environment that is our environment here. So without any further ado, I hand over the floor to Gustav. We have about 45-50 minutes for the lecture, and then we will start with discussion. And yeah, I look forward to both of it.
Thank you so much, Stefan, for this kind introduction. It's great to be here in Berlin with you today. So I'm going to talk today about systematic, transparent, and community-based assessment and quality control of research data. There will be some talk about abstract principles, but I'm going to try to ground this in examples from work that I myself have been involved in and show practically and concretely what we see when we try to find and assess and reuse research data. And so the motivation for this topic fundamentally is, how can we build a science that is cumulative, where we can add one stone to another and expand knowledge by putting together work that is being done by people who have come before us, by ourselves working now, and that can be another stepping stone for colleagues in the future. So let me dive into an example straight away. A piece of work that we did where we tried to find and reuse research data on a moderately large scale. And the topic here is not the main issue, but I'm going to explain very briefly that it's about whether white blood cells of the immune system produce a signaling molecule called interleukin-6 and send it through the bloodstream to the brain at night to make us more sleepy. So the image here is supposed to show some white blood cells signaling to the brain with interleukin-6. And interleukin-6 is well known in the context of inflammation. For example, if we have an inflammation somewhere in the body, then interleukin-6 will be produced, and it will go to a lot of different organs, including the brain, and it can give us fever, loss of appetite, and so on. And ever since 1994, when the first paper came out on interleukin-6 over the day, it has been thought that there is a small variation in healthy humans where interleukin-6 increases at night and makes us more sleepy. So what we can see in this graph is a study of 3 people where interleukin-6 was measured at different times of the day. And they have 2 days and 2 nights, and in the nighttime, the curves go up a little bit. And ever since this study was published, it has been a kind of accepted truth in sleep research that this effect exists. But it's a bit unexpected in the context of the general regulation of wakefulness and sleep, because the brain is usually at the top of the control, as it were, and not being controlled by cells in other parts of the body. So we tried to put together all the data we could find on interleukin-6 at different times of the day. Briefly, we found 43 studies that we could include with 56 different data sets. There were 3 of those data sets where we could find individual participant data, so data from single people and not average over the whole group. There were 36 more studies that had measured interleukin-6 across the day that we could find. We don't know how many there were that we could not find, but 36 that we could find, but that we could not include, in most cases because data were not available. 25 of those studies, and in 7 cases because of a duplication with one of the studies that were included, and in 4 cases data that were that showed very high levels of interleukin-6 that you don't expect to see in a healthy person. Those were mainly from the 90s, when the measurement technique was not as good, so we excluded them. This is the sort of thing that we found when we looked in published papers. The table here is from one paper that decided to show every individual data point for each person and each time point. This is a kind of way to share data openly, even if it's only printed in a PDF format. Unfortunately, very uncommon. But it's a nice way to make data open. More commonly, we saw plots that looked like this on the other side. So this particular plot is from one earlier meta-analysis of interleukin-6 across the day, where the black line shows healthy participants. So it's basically what we were looking for. The red line shows participants or patients with rheumatoid arthritis. And the reason I'm showing this graph is that most of the data were available to us in this format. So we could measure the axes of diagrams, and then we could measure where the data points are. And in that way, we could reconstruct the data set for the group of people that went into it. And so we were able to extract data from many papers. And the result looked like this. This is actually the same plot, but the one on my right is zoomed in. And what we see here is a lot of data points. They are scaled to how many people are in each set of observations. And we can estimate that interleukin-6 drops a bit in the morning, and then it's mainly stable throughout the rest of the day. So we were happy to show a result that contradicted the prevailing wisdom, as it were. And if you're interested in hormones, perhaps you're thinking that it's well known that a hormone called cortisol goes up in the morning and maybe that explains why interleukin 6 goes down. I can't prove it, but it makes sense in light of textbook physiology. So, some reflections now about what this tells us about research data availability and how we can use data. So, first of all, we were able to make a new finding by pooling all the data that was out there and putting it together in one analysis. We came to a result that was not the prevailing idea, even though all of these data were actually out there. Data were often available to us through the published plots. Of course, it would have been better if we had access to tables or spreadsheets or a structured format where we could have extracted the data. Then we would have less error and more precision. What about the data that we couldn't include? Well, they are a problem because they increase the risk of bias. That means that we don't know if the data that we couldn't include would have shown a different result. Maybe data were missing systematically in some way. Maybe data that were never published, for example, showed a flat curve across the day or showed a peak somewhere else. There is no way for us to know this or account for it. Also, it reduces precision. So if we had more data, then we could be more certain about what the curve looks like. Here is a point that I have written rather technically. The lack of individual participant data limits opportunities to model residual heterogeneity. And what this means is that there's a lot of variation in the data that we could not explain with the time of day. This variation is presumably due to something else. Maybe it's random, but it could also be that it's systematic. Perhaps it's related to the participants' sex or age or their body weight. We don't know that. Now, I'm reasonably certain that all of the researchers that collected data actually recorded participants' age and sex and probably their body mass index as well. But those data are not available to us. So now if we wanted to understand how these factors interact with interleukin 6, we would have to go out and collect new data, probably from hundreds of participants, in order to find an answer, even though those data could have been available to us already. And this leads me to a more philosophical point, perhaps. The data that are closed and hidden are a waste of resources. So someone spent a lot of money collecting them, and now we can't use them. And also, it's a failure towards our obligation to the research participants, in my opinion, because they undergo risks and harms in research, which are supposed to be balanced by a knowledge gain. But if the data are gone, if it's not possible to use them anymore, then there is no knowledge gain anymore that can outweigh the risks and harms. In this kind of research with regular venous blood sample, the risks and harms are not that great, but the principle still applies. Let me go one step further here and say the following. When I had done this study, I realized that actually, if I do research, if I collect data from a set of participants and then I put the data in the drawer, somebody else, Stefan, who is sitting across from me, who is trying to understand this research question, he will actually know less if my data are in the drawer compared to if my data had never been collected at all. Because now that they are in my drawer, he has to account for the risk of bias. He has to consider, what if my data showed a particular effect and therefore I didn't publish them because I didn't like what the data were showing, or because the data were showing nothing at all that I thought I could publish. So in fact, generating data without making them known can have a kind of dissipative effect on the cumulative state of knowledge. I wonder if that came out clearly. Perhaps we'll find out in the discussion. Okay, moving on. I wanted to show also some more examples of what we can find when we look for data in the literature. And in this particular case, I was looking for data on sleep deprivation and MRI imaging of the brain. And again, the topic here is not that important. Here are some examples of data availability statements. The first one says that all the data are in the paper, but the paper shows summary results. So what we have here is a kind of evolution where scientific journals have started asking the authors to provide data availability statements. Sometimes they make sense, and sometimes they don't. The first data availability statement does not make sense. The second one contains an error. It says that the data are available in a certain repository, but then when you go to the repository, there's nothing there. By contrast, successful data sharing can look like this. Here is a favorite repository of mine called the Open Neuro repository. And it's simply a screenshot of the latest data set added to it. So I want to point out some characteristics of why this kind of data sharing is successful. First of all, the sharing has been done through a well-recognized, well-functioning repository that takes responsibility for the long-term preservation of the data. We can also see that the data follow a particular data standard. In this case, the BIDS, brain imaging data specification, and it has gone through a validator, an automated check that says that the data follow the specification. This doesn't say, by the way, that the data are particularly good or useful, but it says that the data are structured in a way that follows the specification. There is a unique identifier. So we can be certain that this is one and the same data set. And there is also version control. These are some characteristics that I want to highlight of data sharing. I put the acronym FAIR in the title of this slide as well. It means findable, accessible, interoperable, reusable. You've probably heard of it. It's a very popular acronym in discussions about data sharing these days. Not shown on this slide is that the data also have an open license. Okay, so now I've shown you a few things that I've come across in my various adventures when looking for data. Now I'd like to transition to a discussion about quality control and how we can know what to trust. Here is a perfectly ordinary title page of a scientific paper. It's published in a journal. The journal's name is Scientific Reports. This is a journal that publishes a lot of papers. And we published this one. You can see in the margin that it contains information about when the paper was received, accepted, and published. But we don't know what happened in between those dates. It's not being made public. So if you now want to decide if you can trust what is in this paper, then what can you really base that on? For example, you might assume that the paper has been peer-reviewed. Scientific Reports is a journal that calls itself peer-reviewed, but there is no information about how that happened. You could guess that there have been peer reviewers, but you would have to guess how many of them there were. Was there 1 or 2 or 0? Did the editor do the review themselves? What kind of qualifications did the peer reviewers have? Did they like the paper? Have I made, as an author, any improvements to the paper after seeing the reviews? None of that can be seen in this case because it operates largely as a black box and we need to take it on trust. That is the traditional way that peer review is done. Of course, many journals now operate open peer review, which is much more informative. Nevertheless, what I want to suggest is that we can improve quite a lot on this model by making it more transparent and systematic. In fact, I want to put forward this idea. It's not exactly unique. Many people have been proposing for a long time a move from communicating science in the same way as we did before the internet, with a printed journal article that is static, that is difficult for machines to read, that contains relatively little information about the raw data, where quality control is opaque, to instead communicating science by putting out digital research objects that can interact with one another. Something like this. You could imagine, for example, that if I generate a data set, I put it in a repository, and then I can write code to analyze it, and it can live in a different repository and call on the data that is in the first repository. We can have connections to things like a pre-registration with an analysis plan, and a DMP, a data management plan, that can provide a map to where the data and code are, for example. Interpretations would not have to go in the traditional narrative format of the scientific paper, and reviews and quality control could be open. So again, this is not something I have thought out on a Sunday morning. People have been arguing for this for 20 years at least, but so far it hasn't happened very much yet. And I'll come back to a possible theory of change towards the end. Now I want to go into this box, review and quality control, and make some suggestions for what could go in it if and when we manage to find a way to do it openly. So for the purposes of something that could be checked systematically, what could we mean by research quality? Well, here are some suggestions. Whether the report in the end is consistent with what the plan was from the beginning. That is, if the research has been pre-registered, so the researchers have written a plan, it is entirely possible to check if they have also followed it. We could check computational reproducibility, whether someone else can analyze the data and receive and arrive at the same results. We can check various risks of bias. I'll go into all these points in a bit more detail in a moment. We can check internal consistency, by which I mainly mean obvious errors in reports. And we can also try to throw anything at it, basically. Ask an expert to find any problems that they might encounter. Now I'm going to highlight a few examples of how this might possibly look. So here is an example when it comes to reviewing whether papers follow a pre-registration. There has been quite a lot of work comparing pre-registrations and published papers, and by and large it has shown that there are often discrepancies. In this study, in which I was involved a bit, it was led by Robert Thibault, we tried an intervention. We partnered with some scientific journals and tested what happens if you put in a reviewer whose only job it is to check if the study is consistent with the pre-registration. And we did this as a feasibility study, basically to test if it could reasonably be done. And briefly, we had just over 20 papers. There were important discrepancies in about a third of them, and most of them were addressed satisfactorily after the paper had been through this discrepancy review. And this suggests that this could be done on a larger scale. Importantly, the review here occurs before the paper is published, which I think is a good time to check things. Although I'm also in favor of post-publication peer review on a larger scale. I'm not going to go through all these numbered points. They describe what it was that we looked at in this review. Here's another example. I was involved in a study led by Tom Hardwick, where we tested computational reproducibility. In this particular case, we had a sample of papers from a journal called Cognition. This journal introduced an open data policy, and therefore it was possible to get the data sets from a bunch of papers. We had 35 papers, and about a third of them, when we got the data and followed the methods as described, we could reproduce successfully straight away. For the remaining 2/3, it was not possible to reproduce them. And so we asked the original authors what they had done. And then for about another third, with the author's assistance, we could get the same results again. For the last third, it was not possible, even after the authors had been in touch helpfully, to reproduce the results. And if I recall right, there were 3 papers where the authors decided to issue a correction because there was something wrong. Just to give an example, I had one case with a data set where I looked at the data file and I scrolled down. It was quite a large spreadsheet, and then halfway through it stopped and the bottom half was missing. I mentioned this just to give an example of how the quality control is being performed today, where it focuses on the main text of a manuscript, and it's not anybody's job to look at the actual data. And therefore, I think in this case, the authors had posted the data as a bit of an afterthought and without much attention to making sure that they got the right file. The other plot here shows what kinds of data that we got inconsistent results for. And basically the point is that it was all kinds of statistics. So on the x-axis here you have the individual papers, and on the y-axis different kinds of statistics. From the top, the number of observations, the confidence intervals, miscellaneous, mean values, degrees of freedom, effect sizes, test statistics like a t-statistic, standard deviations, and p-values. And the take-home message is that the errors were in all kinds of numbers. The overall take-home message is that computational reproducibility is possible to test. It can be done in a few hours by volunteers. It is being done by a small number of journals today, including journals of the American Economic Association, if I got that right, the AEA, the journal Metapsychology, and others, but it has not been adopted on a broad scale. By the way, it doesn't require, of course, a journal. You could very well imagine that it could be done by some other third party or by a support function in a university. Risk of bias assessment. We are moving now into something that is often being done in systematic reviews in medical research. And here is a slide that I got from a systematic review. I forgot to put the reference. It shows some of the categories of risk of bias that are very typically assessed. And they include here how the random sequence was generated. That means when the patients were randomized, can you tell how it was done? Like, did someone flip a coin, or did someone put numbered slips in envelopes and then have a colleague draw the envelopes at the testing site, and so on? And it's important to conduct randomization properly in a randomized trial, of course, in order not to have a bias in the selection of the patients. Here, the point that I want to make is that there are well-developed tools for assessing risk of bias in systematic reviews, but they are typically only used systematically at the stage when someone is doing a systematic review after all the papers have been published. This is something that could be done in an ordinary peer review process if we wanted to, instead of having a totally narrative function where you ask an expert to read a paper and say whether they like it. So it's a kind of hint at a more systematic approach to quality control based on something that already exists. Now for something else that I found. We were doing a review earlier this year, and here are some screenshots from a paper that I was looking at. Again, it doesn't matter so much what this paper was about, but very briefly, it was about the glutamate in the brain of patients with something called exhaustion syndrome. And what you see here in the top block of text is the main result of the paper, which was that glutamate levels were higher among the patients compared to the controls. And then they did something which is called an F-test. And an F-test results in an F statistic, and you often report the F statistic with degrees of freedom. But here, the actual F statistic is missing, and we only see the degrees of freedom. It's the main result of the paper, and yet it's not there, which I thought was quite funny. But then when I read on, it said there is a scatter plot. And then I thought, hooray, if there's a scatter plot, then I can go back and measure the plot and reconstitute the data set and get each of the data points and try to calculate the test myself. And the scatter plots were supposedly in the supplementary information, but when I went to the supplementary information, what I found was figure legends, but no plots. They are missing. And this is, of course, just an anecdote. I can make it slightly less anecdotal by saying that there were obvious errors in about 1/3 of the papers that I looked at for this review. Things like the numbers of participants not adding up. For example, a paper saying that there were 32 participants, of which 16 were male and 17 were female. Obviously, that doesn't make a big difference, but it's a kind of red flag that something else might be wrong if there is an obvious error like that. And of course, what I'm trying to imply here is, again, that traditional peer review, where you ask an expert to read the paper and say whether they like it, is failing to detect these internal inconsistencies that could have been detected if you had instead had a systematic framework where someone was asked to simply see if the numbers add up or not. So what do we want? If we think about what peer review could be, here are some suggestions beyond the content suggestions that I've just made. These are more process-oriented features. I think it would be nice to have a quality control system that is independent, meaning it doesn't rely on myself. It is done by someone who doesn't have an interest in whether my research comes out a certain way or not. So someone that a third party could trust not to have a bias. That means it probably should not be carried out by my university, although that could be one way to do it. It's a bit frustrating to me that when I try to build quality control around my own works, it has to be based on myself in some sense. So a hobby horse of mine is that we still do research as if the actor in performing research is a kind of lone genius. There seems to be an assumption that if I do research, then the integrity of the research is guaranteed by my personal professionalism. Now, speaking for myself, I make errors all the time, and therefore I try to get someone else in my group to double check everything. I try to have a colleague who reads my code and who runs it again to make sure that they get the right results. And I do this because I know that I make errors, and I have made some very silly errors that have gone out. But there isn't a third-party service that I could hire. So if I ask Stefan to look at one of my papers, then he will have to trust my process. I can say that I have another person in my group who has reviewed the code, but I can't get a kind of stamp of approval from an independent third party that they have looked at it and verified that it works. Secondly, it would be nice if it were systematic. So Stefan, in this case, again, as a third party, could know that a review has been done according to certain steps, like someone has checked if the data are there, and if the data have the right number of observations, and so on. And transparent. It would be nice if a third party could read the report and see what has been checked. It would also be nice if we had a way to assess many different kinds of scientific objects. So peer review today is very much centered on the traditional reports, of course. I believe we need to develop field-specific standards for assessing different kinds of data. And this is an area where lots of things are happening that I don't have really the scope to go into, but I foresee that there will be a lot of work being done in various organizations that work with research data on developing standards for how to assess fairness, usability, and so on of different kinds of digital research outputs. It would be nice if the system were flexible, modular, and portable, by which I mean if it can be done by different people in different contexts. Perhaps we could have a systematic screening tool, for example, for research data that could be used by a journal or a publication platform or by a third-party service. It would be nice if it could be automated. I don't think that's possible beyond certain basic things. And finally, having a community basis is, I think, essential for reasons that are to do with the social nature of how we do science. Anything that we want to try to push forward needs to be accepted by the community in which it's supposed to work. Okay, and now I'm moving further into the atmosphere with some suggestions for what the world might look like if we had a well-functioning quality control system for research outputs. And I wrote it like this partly because I got, and now I'm going on a small tangent, I got a small research grant to actually try to build something like this, but then the money disappeared because it came from something called the FTX Foundation, which was donated by this crypto billionaire whose entire business went up in flames about 3 weeks ago. And this happened before they had paid out any money. So the grant is gone, but the vision remains, and hopefully I can get someone else to fund some of this work. So what I want to try to do is to build a quality control framework run by professionals. This is important. So not run by researchers in their spare time, which is how peer review typically operates today. If we had this and it were to work, then it would hopefully lead to better incentives for researchers and universities and other organizations to output research that actually meets quality standards, rather than meeting arbitrary standards for publication in certain scientific journals. We have a kind of prestige economy in science where my standing as a scientist depends partly on whether I get published in journals that have high prestige, and then I can borrow some of their prestige. And then if I have very high prestige, I can lend it back to the journals. If I have a Nobel Prize and I publish in a certain journal, it will raise their profile and so on. But if we can find a way to create metrics that are better aligned with the actual quality of research, maybe that could be a way for us to escape the prestige model. It would also enable grant funders to check if their money goes to research of high quality, and hopefully that could lead to more rational allocation of grant funding. And lastly, when strange things appear in the literature, someone would be there to check it. One example there is from the pandemic. Early on, there was a study of hydroxychloroquine for treating patients in the hospitals with COVID-19. This was in the early days of the pandemic when everyone was scrambling to find treatment options. And this initial study led to very many people being treated with hydroxychloroquine in hospitals around the world, but it was found to contain important errors, which were detected by volunteer post-publication reviewers and posted online on the site PubPeer, where people often report discrepancies in science. But I think if there had been some kind of a trusted service for checking data, it could have been detected sooner and could have had a larger impact and would have saved many patients from receiving treatment which was unnecessary and not helpful. Just to give a small motivating example for that last point. Finally, how can we improve incentives for scientists to output quality work rather than output papers published in high-impact journals? Here I need to highlight the recent initiative by the European Commission to put together a coalition of partners around Europe in a kind of top-down approach to reform the way research is being assessed. I think we need to meet this top-down approach with a bottom-up experimentation and creation of new ways to assess research that can be tested during the 6 years or so that this coalition is expected to work. Now is the time to do more experiments, find out what works best, and then inform this top-down work so that now that people have come together to actually align on principles, that they can find the right principles to align on. So for a long time in discussions of reforming assessments, the major counter-argument has been, sure, maybe it would be a good idea, but we can't act alone. We are only one university, or we are only one funder, and we have to not deviate too much from what the rest of the world is doing. Now there is finally concerted action, but we don't know what it's going to be, and we need to create more options and more trials, more evaluation in the coming years. I'll skip to this and say thank you for your attention, and I look forward to the discussion.
Yeah, thank you very much. We now have questions, of course. Thank you for the insightful and thoughtful and I would say also innovative presentation that touches on different topics. I'm looking at Sibylle. Would you want to jump in, or should I first, or how do you feel?
I can start with the question first. Thanks a lot also from my side for for your insights into your vision and your experience with adding more quality control to research. And I think it was very illustrative examples as well, which made it very clear easy to understand. You mentioned several measures for improving quality control during the publishing process, or in the general context of a publication. Are there any certificates in your discipline? I come from the humanities, so I'm not really into the or my knowledge on science is not by far not as large as yours. So are there any certificates ensuring quality control in open publishing, or in, for example, repositories like only for data publication, things like that? So publishing data independently of journals.
That's a great question. I think the closest thing might be badges for open science practices. They have become somewhat popular in the last years. There are about, I think, 80 journals who issue badges that have been kind of defined by the Center for Open Science. Badges for open data, for open materials, and for pre-registration. There are also other journals who offer badges of their own that are related. Most often these badges are awarded just on the basis of authors self-certifying that they meet the criteria, which in reality they often don't. Nevertheless, I think these badges may have a kind of signaling quality, a symbolic significance. There has been one randomized experiment with open data badges, which showed that they did not increase the rates of data sharing in that setting where they were tested. So I think that's my way of trying to answer your question with a yes.
Okay, thanks a lot. As regards repositories, there are several like certificates, like trust seal of approval and so on, but they are very, for university repositories, just institutional repositories, they are quite complicated to obtain, and it's a really, resource intense procedure. So so far, at least in Germany, it's still a long way to go to get these certificates for institutional repositories for data publication. So the question of incentives comes up again in the context of data publication as well. So I was just thinking, where would repositories be in your vision?
Repositories are very central. I would prefer to see a world where we don't need the scientific journals anymore. I think we have perfectly good open platforms already that we could use if it were not the case that we have tied the journals to our systems for assessing merit in hiring and promotion and allocating grants. So repositories are very important. They are fundamental. I believe repositories are mostly a kind of problem that has been solved. Naturally, this varies between fields, but for most of the fields that I can see, good repositories are available, if I now speak very broadly, and that also meet high quality standards. The core trust seal, I think, is a good repository standard. We have started an institutional repository at the Karolinska Institutet, where I am based, and we've gone through the certification process. Notably, it only assesses features of the repository itself. You could still publish data in it that are of high quality or low quality, though there are some minimum requirements for the metadata that describe the data and so on.
Stefan, maybe your questions?
So I actually, this is just the start, and I hope there will be time for other questions, which I find more exciting. But still, I need to understand two more technical aspects about the examples you mentioned. And it is about the very first thing, the interleukin and sleep study. And I mean, first of all, you said that most of the studies that you meta-analyzed actually did not confirm the original finding. So the overall finding was different. So were they then kind of labeled as negative or null results, or were they always labeled as a positive result of a different kind? And more importantly, I'm really curious, how is it possible that you said there were so many studies which didn't even have data available, even though by data availability, you said the standard extremely low by reconstructing data from figures. So how can there be no data available in figures?
Yeah, so it's quite fascinating to go through a body of research and look at how it has developed over time. In this case, 20, 30 years, and to see how people build on earlier claims. And indeed, as you suggest, many studies claim to find a finding that was consistent with interleukin-6 increasing at night, even if they found something like it increases a little bit at 8 in the evening. Yes, we label it as consistent with the idea that it increases at night, or it doesn't even increase at night, but it decreases a bit in the middle of the day. Again, consistent with the theory, and so on. But once you actually put all the data together in the same model, it shows that it's not consistent with that theory. And to your second question, indeed, we set the bar low, but many studies didn't clear it. So there was one single study that gave an explanation and said the data are not shown because the result was not statistically significant. In all the other cases, they don't say why. And sometimes they showed some data, but so aggregated that we couldn't use the results to assess the difference even between 2 points during the day. There was one, I have a favorite story that I can tell about what this might look like in reality. It shows something about how data go to die. It turned out that one of the largest studies that we could not include had been done at our own department a number of years ago. So I went to talk to the professor who was in charge, who had retired, but he still had an office, and he still came in one day a week or so. And I managed to catch him, and I told him, we are very interested in your data set because it's one of the largest data sets that exist on this question. And he said, that's great. I'm very happy to share it, but you have to talk to my colleague who worked with the data, and he has moved to Germany. So I got a hold of this guy, and he said, I'm very happy to share the data, but they are on my old computer, and I don't know how to start it. And so the data probably exist in some sense. They may be sitting on a shelf somewhere in Germany, but we still can't access them. So they are functionally dead. And they are, I mean, I think there's lots of data that are in a kind of time window like this where we could potentially salvage them if someone wanted to put in the effort. And if we don't, then they will become permanently lost.
Any other questions? Otherwise, I would just ask a question, or maybe just interpret it. In Germany, we have this TÜV. We have this organization where you have to take your car to, you have to pay for it, and they give you like a stamp. Okay, your car can go for the next 2 years because it has like technical requirements. Is that what you recommend for research data? There's like an external service provider who will check like certain quality criteria, the one you have listed.
Yeah, I think so. And actually, so that's a quite nice example. I've quite recently, I heard a very senior person who he was the outgoing chair of the Royal Academy of Sciences in Sweden, who said that what is so good about scientific progress is that over time you can tell which findings stand the test of time. And I thought, what other area of human activity could you apply that to? I mean, if you had a car factory, would you say that the great thing is that if the brakes don't work, then people won't drive this car anymore, and only the good cars will still be on the road? Whereas I'm thinking, why can't we have, first of all, someone that checks if the brake works before the car leaves the factory and comes out, and secondly, a third party that checks if they are still working. So I really like that analogy. And again, I think this is my long way of saying yes.
As I'm not an expert in this field, but you said earlier, Sibylle, that these kind of certificates that I would say are kind of comparable to this are quite resource intensive to be implemented in institutional or university repositories. So I think that, as you say, that will be for a long time. Who else could, in the meanwhile, jump in and say we can at least for some disciplines provide some of these services? Do you see any actors? Do you discuss this maybe in the Research Data Alliance or in the European Open Source Cloud? Or maybe, Sibylle, do you know anything like this quality issues being brought up by the NFDI consortia that are now taking off their work, like starting their work?
shall I try to answer? Well, the NFDI, I'm sure the concept is known to you, Gustav. It's the National Research Data Infrastructure movement, you could say. It's a big funding project of the two largest German funding organizations to enable or to empower research data management dependent on disciplines. So there are consortia to develop standards and technical infrastructures for fair data management. So these, yes, these are meant to step in here, but it still will be a long way. For the time being, yes, there are also, for example, for the fairness or the standard or status of verification of repositories or of research data objects. There are web tools like Fuji. I'm sure you know it, Gustav. These are on a, well, I can put the link in here. These are quite helpful also to check the status of openness for institutions, for their repositories, for example, or also for yeah, any kind of data publication online. But I wouldn't be able to answer the question if the NFDI will be the one who will be something like a clearing point for quality data. I'm sure they would say, yes, it's their aim, but we don't know enough right now when and how this will be obtained. So.
Thank you. You want to also respond, or to like?
I can very briefly simply indicate my agreement. The debates I'm seeing in research data circles are circling a lot around data stewardship, data verification skills, data librarianship. There is a lot of talk about how we can build a kind of research data management workforce that can help researchers make their data fair and open, but there's relatively little discussion about the kind of quality control that I've been talking about today. I mean, that's why I want to put it on the table. There's a lot of things going on in other fields, I'm sure, that that I don't fully see, and of course I'm very happy to find out things that I don't know.